Hi, this is Dr. Shanmugapriya, Professor and HOD of Biochemistry at Government Thoothukudi Medical College. I am here to discuss about the significance of glycolysis. The purpose of the session is not actually to just discuss the significance of glycolysis, but to let you know the significance of conceptual learning. There is a huge difference between the effect of factual learning and conceptual learning, and I would say conceptual learning is the only key to learn a subject so that you are able to apply it for your clinical practice. Yeah, I'll give you a sample of it in this presentation. So first, glycolysis. We know, as the name indicates, glycolysis is glyco plus lysis, right? So it's a process in which you split the 6-carbon compound called as glucose to form two products. And we know that the two products which we get will differ depending upon whether the process occurs aerobically or anaerobically. When the process occurs aerobically, the products formed are 2 molecules of pyruvate and 7 ATPs. When the same process occurs anaerobically, products formed are 2 molecules of lactate and 2 ATPs. So let me repeat this. Products of aerobic glycolysis would be 2 pyruvate and 7 ATPs. Products of anaerobic glycolysis would be 2 lactate and 2 ATPs. So if you are asked what is the significance of glycolysis, please don't answer that it's a pathway which can generate ATP, because we have n number of pathways happening in our body which can synthesize even more number of ATPs than what glycolysis can. So that is not the significance of glycolysis. If you are asked what is the significance of a pathway, it means they are asking you what is unique about this pathway. So the unique feature of glycolysis is that it's the only pathway which can generate ATP even in anaerobic conditions. Do you understand this? It's the only pathway that can generate ATP even in the absence of oxygen. So your next question should be, when we are all aerobic organisms, human being is an aerobic organism, right? So when we are all aerobic organisms, then why do we need a pathway which can generate ATP even in the absence of oxygen? If you ask yourself this question, yeah, the most probable answer that your mind will tell you is, okay, what if I'm hypoxic? What if I don't get enough oxygen? I might be dependent on glycolysis would be the first answer. But that's not the right answer because we are not speaking here about abnormal conditions. We are now speaking about normal conditions. Even normally in your body, there are few cells which are dependent on anaerobic glycolysis to generate the required ATP. And those cells include RBCs, yeah, retinal cells, corneal cells. RBCs, retinal cells and corneal cells do not have mitochondria. That we all know, right? They don't have mitochondria, which means they cannot undergo aerobic metabolism. So these cells are dependent on a pathway to generate ATP in the absence of oxygen. For these cells, glycolysis is indispensable. So please have this in mind. Glycolysis is indispensable in RBCs, retinal cells and corneal cells because they don't have mitochondria. Similarly, white muscle fibers. So what is the difference between red muscle fiber and white muscle fiber? Red muscle fiber is red because it has got myoglobin. And myoglobin, remember, is the storage form of oxygen. So what is the difference between hemoglobin and myoglobin? Hemoglobin is the transport form of oxygen. Myoglobin is the storage form of oxygen that is available in red muscle fibers. So why do you think nature has provided only muscle fibers with a storage form of oxygen, that is myoglobin? Your logical question to yourself should have been, why not for hepatocytes? Right? Why not for any other cells? Why do you think nature has provided only a muscle with a storage form of oxygen? So the answer to that question is, muscles, when they contract, yeah, when muscles contract, what happens to blood vessels which are getting into those muscle fibers? blood vessels get constricted. So muscles get detached from a supply of fresh oxygen exactly when they need it. Right? Only when they contract, they need oxygen. But when they contract, blood vessels get constricted, they don't get a fresh supply of oxygen, which means they are dependent on a storage form of oxygen, and that is myoglobin. So red muscle fibers which have myoglobin, which is a storage form of oxygen, can undergo aerobic metabolism. Whereas white muscle fibers which lack myoglobin will have to undergo anaerobic metabolism. Okay, and if I say white muscle fibers undergo anaerobic metabolism, that's a clue that is given to you that this cell can utilize only glucose as its fuel. This cell can utilize only anaerobic glycolysis for ATP production. So whenever you hear the term an anaerobic cell, you can close your eyes and say this cell can use only glucose, this cell can use only anaerobic glycolysis for ATP production. Whereas red muscle fibers, I said it has got myoglobin, which is a storage form of oxygen, so it's going to undergo aerobic metabolism. And if it's aerobic metabolism, a cell has got two choices of fuels. One is glucose, the other one is fatty acid. Yeah, all of you, please remember this. Glucose can be used both aerobically and anaerobically. Fatty acids can be used only aerobically. Okay, so if it's an anaerobic cell, the fuel is only glucose. If it's aerobic cell, you have two choices of fuels. One is glucose, the other one is fatty acid. But given a choice, what do you think a muscle would choose? Yeah, given a choice, a muscle or any cell for that matter should choose only fatty acids, because fatty acids on oxidation, fatty acids on oxidation provide more number of ATPs than glucose on oxidation. I'll make it factual. Palmitic acid, for example, palmitic acid on oxidation provides you 106 ATPs. Whereas glucose on aerobic oxidation gives you 32 ATPs. So what would a cell choose? Fatty acid. So what is the preferred fuel for your red muscle fiber? It is fatty acid. What is the preferred fuel for your white muscle fiber? It is glucose. Okay, and that is why red muscle fibers store fatty acids more and more. In the form of triacylglycerol and cholesterol ester. And that is why we always say, do not take red meat. Suppose you come across a patient with hypercholesterolemia, hypertriglyceridemia, and the person naturally would ask, what would be the dietary recommendation for me? What changes should I make in my diet to reduce my cholesterol or triglyceride? Your answer should be, I know you must have heard of answers like avoid red meat. So why are they asked to avoid red meat? Because red meat is full of triacylglycerol and cholesterol ester, because its preferred fuel is fatty acid. Okay. So what are the cells for which glycolysis is indispensable? RBCs, retinal cells, corneal cells which do not have mitochondria, white muscle fibers which do not have myoglobin. Okay. So that's about the significance of glycolysis. So why do we have to know about significances of glycolysis? It is for you to understand what happens whenever there is an inborn error of metabolism related to glycolysis. For example, Tarui's disease. Don't worry if you don't know this disease, memorize it now. Tarui's disease is otherwise called as glycogen storage disease. Storage disease type 7. And the enzyme which is defective in this condition is phosphofructokinase 1. So suppose you are asked, what do you think will be the manifestations of Tarui's disease? And you're given a fact that's a defect of phosphofructokinase 1, the rate limiting enzyme of glycolysis. Your answer is going to be very simple, because glycolysis is indispensable for RBCs and for white muscle fibers. Okay. Now my question to you is, why do you think RBCs need ATP at all? Yeah, since the beginning of the session, I've been telling you RBCs don't have mitochondria, but they have to produce ATP. So they're dependent on anaerobic glycolysis. So they're dependent on glucose as their fuel. Agreed. But why do you think they need ATP at all? It is not to carry oxygen, because whenever we say RBCs carry oxygen, it's not like it's lifting oxygen and it's going. It's just that oxygen binds to Fe2+, and then circulation carries the oxyhemoglobin to tissues wherein oxygen is diffused. So it's not a mechanical work which RBC is doing when you say oxygen is transported by RBC. So that's not what ATP is used for in RBCs. RBCs need ATP for the functioning of sodium potassium ATPase pump. Yeah, they need ATP for what? Sodium potassium ATPase pump. So what does this pump do? Anybody? You can think of an answer for this. What does this pump do? This pump pumps out 3 sodium ions, okay, and it reciprocally takes in 2 potassium ions. Okay, what does this pump do? The pump pumps out 3 sodium ions. When I say out, it's out of cell, which means it's pumping it into the circulation. And it takes in 2 potassium ions. And you know your intracellular fluid is already rich in potassium and your extracellular fluid is already rich in sodium. So it's moving both these ions across, it moves both these ions against concentration gradient, for which it needs energy. And that energy is obtained by hydrolyzing ATP. Okay, so if you're asked what is the ultimate aim of this pump, why does it pump out sodium ion, your answer should be it avoids sodium from getting accumulated within the cell. Because if you allow sodium to accumulate within a cell, sodium being osmotically active, it would attract water. Yeah, it's osmotically active, so it attracts water and the cell will swell and the cell will rupture. So which pump avoids all your cells from undergoing rupture? It is sodium potassium ATPase pump, not just in RBCs, every cell for that matter. Yeah, that pump in RBCs need ATP, and that ATP in RBCs is provided by anaerobic glycolysis, for which it uses glucose. Okay, in that case, tell me what is the effect of Tarui's disease or type 7 glycogen storage disorder caused by the defect of phosphofructokinase 1, which is the rate limiting enzyme of glycolysis. ATP will not be produced in RBCs, so sodium potassium ATPase pump fails. Sodium accumulates within the cell swells and the cell ruptures. An RBC rupture is what we call as hemolysis. So it presents as hemolytic anemia. So tell me, what is the principal presenting feature of glycolytic enzyme defects? It's going to be hemolytic anemia. And when I say hemolytic anemia, what will be the clinical picture? Hemoglobin concentration will be less, and that will be accompanied by reticulocytosis. So hemoglobin concentration is less, and that's accompanied by an increase in reticulocyte count. And because within RBCs we have lactate dehydrogenase enzyme, LDH activity will be high in the circulation. And whenever there is hemolysis, heme will be oxidized by heme oxygenase enzyme to form biliverdin, and that becomes bilirubin. And this bilirubin should undergo conjugation, but liver has got its own maximum conjugating capacity, which when exceeded, unconjugated bilirubin concentration increases. Yeah, there will be an increase in unconjugated bile pigment concentration. So all these will be observed as a manifestation of hemolytic anemia in Tarui's disease. Okay, now not only RBCs, even white muscle fibers get affected in glycolytic enzyme defects. And when white muscle fibers get affected, logically it should present with exercise intolerance. But all these glycolytic enzyme defects appear in the neonatal period. It's not going to wait for a person to grow up, to start walking, to start running, for a person to come and complain to you saying, I have exercise intolerance. Okay, so it appears earlier. All these manifestations appear earlier. So when the child, when there is a newborn, you won't be able to find out exercise intolerance by taking a history. So you perform a biochemical investigation and you find that CK-MM level is elevated. Yeah, we know CK are of three isoforms, MM, BB and MB. MM is present in skeletal muscle. So that isoform level is elevated in circulation, denoting that in this condition, white muscle fiber is also affected. Okay, so if you don't understand this concept, if you don't understand all these concepts, if you try to just memorize, okay, Tarui's disease, a defect of phosphofructokinase 1 and it presents with hemolytic anemia. In your exam or even in clinics, when you come across such a patient or you're given a clinical scenario and asked to diagnose, okay, you will think of glycolytic defect. Maybe it presents with hypoglycemia. Maybe it presents with hyperglycemia because it's a glycolytic defect. It uses glucose. Yeah, even if you stretch your imagination, it would go only till then, right? That is why factual learning never helps. Don't memorize clinical features of any of the inborn errors of metabolism. Try to understand the concepts behind it. Try to understand the reason behind it. So what are the manifestations of any glycolytic enzyme defect? It can be phosphofructokinase 1 defect, which is Tarui's disease, or it can be a pyruvate kinase defect that's immaterial. All these glycolytic enzyme defects would present as hemolytic anemia, characterized by reduced hemoglobin concentration, increased reticulocyte count, increase in LDH level, increase in unconjugated bile pigment concentration. And along with that, because white muscle fibers also get affected, CK-MM level will be elevated. Okay. Now coming back to utilization of fuels by all these cells. So what I have already told you is, about RBCs. And we know for sure RBCs use only glucose. Okay, forever it uses only glucose. Similarly, retinal cells and corneal cells and even white muscle fibers. Okay, now how about cardiac muscle fibers and red muscle fibers? Both cardiac muscle fibers are known for their mitochondrial content. Red muscle fibers are known for their myoglobin content. So what metabolism do you think they undergo? Aerobic metabolism. And if it's aerobic metabolism, I've given you there are two choices of fuels, glucose and fatty acids. But given a choice, what would a cell choose? Fatty acid. So what is the preferred fuel for cardiac muscle fibers and red muscle fibers? It's fatty acid. Okay. And what is the preferred fuel for neurons? Neurons. Just think, is neuron aerobic or anaerobic? Neurons, they are aerobic cells, and we know the concept, right? If it's aerobic cell, they can use either glucose or fatty acid, and always I say, given a choice, they choose fatty acid. But in neuron, the case is totally different. Though they can use fatty acid, they don't use fatty acid. Instead, they use only glucose. Reason is, in your circulation, fatty acids never stay alone. Fatty acid is conjugated with albumin. Yeah, fatty acid is conjugated with albumin. For simple reason, all these fatty acids are non-polar and your circulation is water basically. So non-polar fatty acid cannot be soluble in plasma. So to make it soluble, it is attached, it's conjugated with albumin. And anything which is conjugated with albumin cannot cross the blood-brain barrier. So your neurons do not have an access to fatty acid. So neurons have evolved to utilize glucose instead. Do you understand this? So what is the preferred fuel for your neurons? Though they are aerobic cells, they preferably utilize glucose because they don't have an access to fatty acid which is conjugated to albumin which cannot cross the blood-brain barrier. Only in extremes of starvation, neurons start utilizing ketone bodies. Do you understand this? Only in extremes of starvation, neurons utilize ketone bodies. Even in extremes of starvation, they use glucose and ketone bodies in the ratio 3:1. So even in extremes of starvation, if you are asked what is the preferred fuel for neuron, your answer should still be glucose. It is just that it starts utilizing ketone body. Its supplementary fuel becomes ketone bodies because ketone bodies can easily cross the blood-brain barrier. Okay, so what are the preferred fuels for all the cells you can think of? You start with RBCs, retinal cells, corneal cells, white muscle fibers. It would be glucose because they are all anaerobic cells. Red muscle fibers and cardiac muscle fibers because they are all aerobic, they utilize fatty acid. Neurons, though they are aerobic, they utilize only glucose preferably because fatty acids conjugated with albumin cannot cross blood-brain barrier. In extremes of starvation, neurons start utilizing ketone bodies. And even in extremes of starvation, they utilize glucose and ketone bodies in the ratio 3:1. So even in extremes of starvation, the preferred fuel for neurons would be glucose. Okay. So these are the facts which you would understand from one fact related to glycolysis. What is the fact I told you? Anaerobic glycolysis can generate ATP. Yeah, this is how linked your learning should be instead of being purely factual. So don't read an MCQ, don't just memorize an answer. Try to approach all MCQs conceptually. Yeah, that is the basis of our learning app, speed learning app. So let's start discussing few MCQs. You will understand how easy it is to answer these MCQs now. Glycolysis occurs in all except. Yeah, does it happen in RBCs? Yes. Does it happen in neurons? Yes. So can you think and tell me what is the difference between glycolysis happening in RBCs and glycolysis happening in neurons? In RBCs, it is anaerobic glycolysis. In neurons, it's going to be aerobic glycolysis. Okay, but it happens in both the cells. White muscle fibers, yes. How about cardiac muscle fibers? Until cardiac muscle fibers are perfused and until cardiac muscle fibers are oxygenated, they use fatty acids through fatty acid oxidation. Only for any reason there is hypoxia or for any reason there is hypoperfusion to cardiac muscle fibers, they switch over and they start utilizing glucose anaerobically. Okay. So what is answer to this question? Cardiac muscle fibers. The preferred fuel for neurons is, we know that for sure, glucose because fatty acids cannot cross blood-brain barrier. Next question, the preferred fuel for neurons in extremes of starvation is, at any point of time, preferred fuel for neurons would be glucose. Glycolytic enzyme defects present with. Yeah, I told you, right? Glycolytic enzyme defects do not present with hypoglycemia or with hyperglycemia. This is not a feature. Is this a feature? Can lactic acidosis be a feature of glycolytic enzyme defect? No. Without glycolysis, no pyruvate, no lactate. Does it present with hypophosphatemia? No. What does it present with? Hemolysis. A 36-year-old woman gave birth to a term infant by elective cesarean delivery after an unremarkable pregnancy. The 3.4 kg boy was hypotonic and needed intubation at birth. A first blood count revealed anemia. Okay, reticulocyte count was high. Anemia with reticulocytosis, immediately think of hemolysis because in hemolysis there will be compensatory increase in erythropoiesis. So young RBCs will be flushed into circulation. At 5 hours of life, LDH activity reached 5,411 units per liter, which is very high. Unconjugated bilirubin was high. So till this, they have tried telling you that this child presents with hemolytic anemia. CKMM level was high. So hemolytic anemia with increase in CKMM level or skeletal muscle getting affected would be a clue that is given to you that it's a glycolytic enzyme defect. Peripheral smear revealed that it's a case of non-spherocytic hemolytic anemia. Yeah, it's not hereditary spherocytosis, which is the reason for hemolysis in this condition. Pyruvate kinase activity was remarkably low. So that is also given to you. Glycolytic enzyme defect, to be very specific, they have mentioned that's a pyruvate kinase activity decrease. Which of the following explains hemolytic anemia in this condition? So what is the reason for hemolysis in this condition? Without glycolysis, no ATP. So the pump fails, right? So it is failure of sodium potassium ATPase pump. So it's as simple as that if it's a conceptual learning. Yeah, thank you.